Good evening, ladies and gentlemen. If I could have your attention, we're going to go ahead and start the program. My name is Alex Wallace. I serve as one of the co-chairs for the World Oceans Week Committee here at the Explorers Club. And we are on the 5th night of Oceans Week. And this just keeps getting better as far as I'm concerned. Our first program is going to be on Foraminifera with Dr. Beverly Goodman and her team. But before we get started, one housekeeping note. For those of you that have a wristband, when we finish this program, stay seated. For those that don't have a wristband, you can exit this way. We've got to get the room turned around for the next show. So everybody got that? Wristband, stay seated. Anyway, I wanted to introduce Dr. Beverly Goodman, and she'll introduce her team. She's a professor in the Department of Marine Geosciences at the University of Haifa in Israel. She got her start academically at University of Wisconsin-Madison. I believe received a master's at Penn State, and then her PhD at McMaster University in Canada. She is a fellow in the Explorers Club, been a member since 2008. And it's interesting because we select 3 ambassador species every year for Oceans Week. And, they kind of do some of the big names ones that everybody, yeah, I know what that is. But when we suggested Foraminifera, you got a lot of looks. People all of a sudden like, what is that? Well, tonight you're going to find out why we selected it, and it was for a very good reason. They are really cool. They're very important. They exist primarily in the benthic zone. They're everywhere. I even found out a little while ago they're in the pyramids, in the blocks that consist of the pyramids. So they're a source of food, they're a source of building materials, and we'll let Beverly go ahead and start from now. And don't forget, wristbands, stay seated.
Thank you so much.
Here's your clicker.
Great. So talk about calling my bluff, right? So yes, when the topic of ambassadors came up, I popped up with let's do Foraminifera, and shockingly, they selected Foraminifera. So, and you guys came, so good job. The sellout Foraminifera. So I'm going to introduce the subject of Foraminifera, but before I do that, of course, exactly what I just said, that normally when you hear about marine research, you think of the sharks, you think of the whales, you think of the octopus, you think of these sort of critters that, sometimes we call them charismatic animals, things like that really draw us. But of course we know that the majority of the animals in the ocean and on the planet aren't quite so charismatic. So I'm hoping that by the end of this session, you will see why, for me, instead of paying attention to this octopus, you might think I'm looking at the octopus, but I'm probably actually looking at everything that's clinging to that rock between me and the octopus. So that's what I'm going to be talking about. Okay, so let's begin. Forama what, all right? So we're going to work on this. So what is this thing? And we're going to learn about who are they, okay, throughout these different talks today. And most importantly, why, okay? Why in the world did it get selected? Why in the world are we investing our time in working with these animals or organisms? And I hope by the end you'll be one of the insiders on this whole Foraminifera business. Okay, so we should start with a resume, right? Okay, we've got to introduce. They already got selected, okay, the candidate was hired, but I feel like maybe we should review why that choice was made. So, we've got Dr. Foraminifera, and, they got their degree on the Earth, all right, they're an Earthling, and they've been around. You mentioned they were part of the Giza Pyramid. In fact, did a whole bunch of work with a certain Dr. Hook back in the time that they were building microscopes. In fact, whole lot of important things like oil, gas, they kind of made their contribution. They're trying to make reparations now, moving on, diversifying, but that is part of the history, and we need to acknowledge that. And basically, they have expertise in ice age reconstructions, sea level reconstruction. It's ways that Foraminifera have contributed to why we know that there are ice ages at all. We also have used them to understand how the chemistry of the ocean has changed, and yet none of us have ever heard of them. But that will end very quickly. Okay, they're also gorgeous, okay? They are single-cell organisms, if you can believe it. that means that foraminifera actually create these very ornate tests, these very fancy shells, and they're doing it with a single cell. Now, I have more than one cell, and I don't look this good every day, okay? I gotta work at it. But they have figured out over their history, 500 million years, that we find them in the geological record. So they came around on the planet about the same time as coral. Have you heard of coral? Okay, yeah. So, all right, that's okay. That's okay. But anyway, they came around about the same time as coral, and they are very similar in many ways. Okay, they photosynthesize, they have symbionts, they're made of similar material. They're kind of like a miniaturized version in many ways of coral, and so they're also useful for research where the work that's done on foraminifera is also very relevant for coral as well. Now, the image that you see here, which is quite famous, they were also, because they're very small, right, they're the size, basically, the biggest ones might be a couple centimeters, and the pyramids, you'll find ones that are even bigger, single cell, but generally speaking, it's sand grain size and smaller. So that is quite small, but it's big enough that the earliest microscopes could see them. So they have hundreds of years, some of the earliest microscopic organisms that were described and named. So they also have this really, really nice long history. We find them all over the world, and we learned that we could find them all over the world because in the 19th century, one of the most important expeditions ever in oceanography was the Challenger expedition. And they went around the world, and this is one of those wonderful things that I wish it were still this way, where they basically put out an application that said, we want to go around the world and we want to see what's in the ocean. I don't think you could get a grant today if you submitted, such a project. But that's what they did. And one of the big questions was, what does the bottom of the ocean look like? We're still asking this question in many ways, but they really had a lot of debate. Is it cold? Is it dead? Is it, a desert? What is it? And some of the samples that came up, they would get to the ship and they would go through them and by hand draw what they saw through the microscopes. So what you're looking at here, and I suggest to everybody get online, look up Challenger report. It's wonderful. You can find the entire report online. It's beautiful. And these are these beautiful colored drawings from that expedition report that is accessible to everybody. So in this drawing, a lot of what you're seeing there are foraminifera. Here's an example of one of these drawings, one of these beautiful drawings from the Challenger. And on the right, I'm going to put up a nice image from Howard Sparrow from Catalina Island. And this is a photograph, okay, of what that foram looks like. And the little shiny dots that you see around there, actually, they're symbionts. Okay, that's a single-cell organism. And it can even capture prey that is about 1,000 times its size and eat it. So don't think they're like precious and delicate. They are vicious hunters, some of them. And of course, today, we're using all kinds of geochemical analysis and all sorts of advanced techniques where we're able to do isotopic analysis, geochemistry. There's endless things, and we do have conferences just for forams. Granted, we only do it about every 4 years because we only have maybe 500 people there. But in any case, there's a very, very wide range of topics that we can cover with foraminifera. So today what I did, and just more beautiful images, lots of beautiful images, some examples. So today, I've invited speakers who are going to talk about different ways in which foraminifera are used in research. We have, from the left to right here, Akos Kalman, Dr. Akos Kalman, who's now at the University of Miami. He's a postdoc working on a National Science Foundation grant with Professor Sam Purkis. And he is doing a whole wide range of questions from droughts and tsunami issues and climate change, and a project looking at, he's going to talk about research that they're doing about coral, taking care of coral restoration projects and how you can apply foraminifera studies to these kind of environmental questions. We also have with us Alyssa Petrazik, who shortly will be Dr. Petrazik, and she is right now an adjunct faculty at Bryant University and Southern New Hampshire University, and she has been focusing her work on sea level markers and change and also applications within archaeology, particularly coastal and underwater archaeology. And we have a recording from Dr. Edward Reinhardt, who researches foraminifera in order to better understand cave systems in the Yucatan. Okay, so I tried to get a nice wide range of different topics that can be addressed using these amazing creatures. So with that, oh, there we are. Okay, so I'd like to invite Akos to the...
Slide, please. Good afternoon, everyone. Today I brought you two of my favorite examples of application of foraminifera. One of them is to use that for paleoclimate reconstruction, and the other one is for coral reef restoration. But before that, I would like to invite you to my office. This is my office. This is where I sit, usually. You see a binocular. I have lots of samples here waiting to be analyzed. I have a foraminifera atlas, plenty of articles, a coffee cup, of course. It's needed. So all these things, this is a general settings when we look at the forams, when we are dealing with marine geology. I'm a marine geologist, which means I'm asking questions from the sediments. So when I look at them under the microscope of the foraminifera, I can ask, for example, what kind of foram this is. I can ask why this is pristine, why this one is broken, why the other one is abraded, why this one is broken and abraded, why they are colored, what is the ratio between pristine versus broken or abraded forams. So all these questions can come when we look at them under the microscope. And because the forams are very, very ubiquitous in the marine sediments, this allows them to use them for statistical calculations. So everything begins actually with a research question. If you are interested in processes in human time scale, for example, human time scale in 100, maximum 200 years of changes in the environmental conditions in the past, we take the uppermost part, of the sediment, we scoop it in, and then we take it to the lab and we analyze it. Usually we have a visitor who is always interested in, and a fieldwork, which is always so much fun. When we are interested in going a little deeper, going back in time, then we take a sediment core. Here you can see a push core because we push it into the sediment. This is approximately like 30 centimeters long, and we can get information on past environmental conditions for the last couple of hundreds of years. And if we want to go a little more back in time, first of all, we need a longer sediment core, and we need more than just a human power. Over here, we use a hammer, a pneumatic hammer in this case, to go deeper in the sedimentary core. If we are lucky, we can have a sediment core which looks like this. Okay, here you can see different colors, you can see different grain size, maybe you can see biological inclusions. Altogether, we see that there were changes in the environmental conditions in the past. Or for example, here, where I can see, where I can see that the uppermost layer, it's a light brown layer, lays on the top of these darker layers. And another example, which shows that the top of the sediment core, the top 11 centimeter is remarkably different from these underlying layers. It's a little bit more yellow. I don't know if you can see that. Coarser, and this one is finer and a little bit darker. So let's see what the foraminifera have to say about this. Okay, so this is the last core that I just showed you. Here you can see this different section, horizon. This is the top 25 centimeter of the core. And what we did in the lab is we counted the total number of foraminifera in every centimeter. So this is a 25 centimeter core, 25 samples. Here it comes. Are you ready? Okay, so you see that the values fluctuate here a little bit, but then a huge increase exactly where the new horizon begins. Okay, this fluctuation was around 500 foraminifera per milliliter, and then something happened, something huge, something that created conditions which favored the living conditions for the forams. So suddenly, from this 500 foraminifera, there was a 3 times increase and reached around 1,500 forams. We don't know exactly at this point yet what happened. We just know that it was recorded and something major happened that resulted in an increase in the foraminifera numbers. When we have a sediment core with horizons, we are lucky. We are absolutely happy about it. But it can also happen that we go out, we take a sediment core, and then for the first sight, it looks completely homogeneous, right? Pay attention, this is a 3-meter long core, 300 centimeter, and again, let's see what the forams have to say about it. Okay, so it's not homogeneous at all. There are a couple of things you can read from here. You see that the foram numbers start very low. Then there's a long-term pattern throughout the entire core. Okay, it starts increasing. Then it reaches its maximum abundance at around 150 centimeter. Then gradually it decreases towards the top of the core, which is the bottom of the sediment of the seafloor. Okay? Over and above this one, you can see if you look, for example, at this section looks a little higher than the previous section, but these numbers, deviation is the same. So what this is trying to tell us, for example, even here, you see at the highest peak of the foram abundances, but you can easily subdivide it into 3 different regions based on these deviations. One of them, this one, there's a little decrease. Then there's an upper section with higher increase. So what this is trying to say is that even though we have this long-term pattern, probably a millennial timescale, changes in the environmental conditions, locally, for a couple of hundreds of years, the foraminifera is trying to tell us that there's a local something like shorter cycle where the conditions probably remain very, very similar. So because climate and sediments are related, we can say that the foraminifera are excellent bioindicators for paleoenvironmental and even paleoclimate reconstructions. So here we arrive to my current research. I'm working at the University of Miami with Sam Perkis, Alex Humphreys, and amazing students from the university using foraminifera to detect past coral algal phase shifts. And I have to say this is urgent because unfortunately we have a problem. The problem is that up to today, almost 50% of the coral reefs died already in the last few decades. And even though targeted restoration efforts can reverse this process, there's a point when it becomes extremely expensive or even sometimes irreversible, impossible. So the challenge of my research I'm working right now is using foraminifera to identify which corals would benefit the most from targeted interventions. Is there a way to differentiate where is this point where we can channel all our efforts instead of like burning money to coral reefs which are either naturally depeporate or they are already at passing the stage. So this is how we arrive to my original site, Vieques, in Puerto Rico. It's a tiny island in the middle of the Caribbean where the coral reef started to degrade as early as the 1970s. So again, this is at the island. And the research team collected sediment samples, surface sediment samples, offshore the island in 10 different transects in 2004. And we analyzed that for foraminifera abundances. This island was occupied by the US Navy for a pretty long time, and they used it for weapon training and let's just say for other military activities. But because of that, the areas were documented by aerial photography. They had to check the result of their testings. So we used that for the foraminifera research, and what we did from all these sites that you see here, we categorized them into benthic habitat types that was monitored by the aerial photography in the course of 80, 90 years. So you can see, for example, the first example is we categorized the stable sand. From 1959, you can see that the sample location, this is just an example, but all of them were categorized them into, you will see the groups. Nothing has been overgrown throughout the years. Okay, so we call it as a stable sand. Why this is generally important? Because different foram communities populate different habitats in the marine environment. So if we can see that and distinguish them, we can actually see which reefs have been shifting or which ones were stable throughout the last couple of decades. So our next category is the stable SAV. SAV stands for submerged aquatic vegetation, which is usually a common word to describe seagrass. And you can see here, this is the early picture, 1936. This very dark, patchy, most probably seagrass, and it was like actually confirmed from the sampling that was in 2004. It was densely dominated, then later on sparsely, but has always been seagrass. We have a very important third category, the stable reef sand. This is the reef, and you can see it has always been a reef. And you also have the transitions in between, between a sand and this SEV category. Over here in 1936, you can see maybe just started to overgrown by the SEV, but presumably that has always been sand-dominated regime. And then you can see the SEV all the way. And the most important category for us, the transition from reef to macroalgal dominance. Okay? Over here, there's no patch, but then this patch started to be darker and darker as we moved towards the present or the sample collection. From all the vehicle samples, we identified altogether 41 foraminifera genera. This is just a list, but I'd like to pay attention for you for this. This is 1% abundances forams, and this is more than 1%. Foraminifera, so this is the second, third league players. These are the hardcore foraminifera, the ones that dominate in the sediment. And we use the statistical tool, get ready. We call it the principal component analysis. What it does is actually is finding structure and relationship between all these 41 genera and the 5 distinct categories of the benthic types. Okay, and this is the result that we got. I'd like to draw your attention that all these categories are distinct. There's no overlap in between. Every point marks a sample location, and the color represents what kind of benthic habitat it was. So this is sand, the yellow. The green one is seagrass. We have the blue one, the coral, and the transition in between. And here we have just 2 points for the transitions. So this is great, but we are halfway done. If we want to identify phase-shifting corals fast, we need much more foraminifera than just 41 genera. So that's exactly what we did. We decreased the number of foraminifera genera, and we were hoping and crossing the fingers that this separation still remains the same. So if you keep your eye on that, let's wait for the GIF to start again. So we started at 41. At 16, it still remained the same. There's a little changing. 8, 6, 5, 4, it's still 3, and 2, it's already not good. You see? The groups overlap, it's not good. So we have to go one more foraminifera genera back in order to describe benthic habitat types. Okay, so this is the 3 genera, very nice distribution. And these 3 foraminifera, their names are very good, get ready. This one is called Quinquiloculina. This is Amphistagina. And the third one is Archaeas angulatus. Okay, so what we see here is that starting from this left, the sand region is pulled by the Quinquiloculina abundances. It means it has high abundances of Quinquiloculinas, and it has low abundances of these other 2, Amphistaginas and Archaeas. As we move to the right, the number of the percentages of Quinquiloculinas decrease, and we have higher abundances of these two. And when we get to the coral reef sections, basically we don't have quinquilloculinas, and we have just these two foraminifera, which are very similar to the coral reefs that Beverly mentioned. They are photosynthetic, they have photosynthetic symbionts, and they are particularly sensitive for environmental changes like the corals. So this is excellent. There's no overlap between the categories. So we can translate this two-dimensional graph into one dimension. You can see there's still no overlap between the categories. And using the boxes and these whiskers, we can actually translate it into a more fancy version, an entire spectrum. And we call it the foraminifera assessment of sediment transitions, a fast spectrum, that we can identify just using the forams what kind of benthic habitats they lived in. Okay? We had another sample collection, apart from Vieques, from the entire Caribbean region. We have samples from Miami, of course, many samples from the Bahamas, a few samples from Jamaica, a few samples from Colombia. So we can have, by using them and calculating these fast values, how well the coral reefs are doing. And very conveniently, all these samples and calculated values fall within our spectrum. And this is saying that the Caribbean reefs are not doing well. And we can actually select which are the regions, which foraminifera, which locations need the immediate attention for restoration efforts. And based on that, I have the last slide for you. The two examples from the Bahamas. One of them is Quesal Bank, and the other one is Great Inagua. Okay? With the blue polygons, these are the actual coral reefs. And this heat map represents the benthic habitat types based on the foram index and spectrum. Okay? What we see here is both of the regions are transitioning. But in case of the Quesal Bank, where you can see this is it's in a very remote location, located in a very distant location, probably exempt from direct human activities, but still the corals are not doing well. And this is because it's a naturally changing, it's a dynamic environment. It doesn't give very well conditions for the corals to expand. Okay? On the other hand, Great Inagua, where you can see high level of alteration of the coastline in the inland, there's a town here, there's an airport here, there's a company that makes salt and then induces runoff more and more nutrients to the coral reef regions. You can see these alterations happened in the near past. And these fast values indicate that we are very close to pristine conditions. This gray, the blue one is the coral condition, gray one is like very close to that. So based on the foraminifera, I would say I wouldn't burn a single money on trying to save this Quesal Bank situation because it's naturally changing. And I would channel all the efforts and restoration projects to Great Inagua, for example. With that said, thank you very much. So it's my pleasure to introduce Alisa, our next speaker. Please come.
Thank you so much. I'm so happy to be here. No, get my slides, please. Great. Okay, like many people in this room today, my first love is the oceans, but my second love is archaeology. And given the opening and the introduction of connection between foraminifera and the Giza pyramids, figured talking about the applications of foraminifera to archaeological research would be a nice place to go. While I bet most of the people in this room have probably never heard of foraminifera until today, I would wager you probably have heard a lot about archaeology. So today I'm going to be giving you a brief overview of some of the applications of foraminifera to archaeological research, more specifically focusing on their applications to the identification of shell middens, ceramic research, and ancient harbor studies. And mostly we're going to be giving examples from other people's work, but at the end we're going to dive into a little bit of my own research. So for all of you who are a little new to Foraminifera, maybe a little bit overwhelmed at this point, the two things to kind of keep in mind are different species of Foraminifera are indicative of different environmental conditions. So certain species live in low energy environments, certain species live in high energy environments, some live in deep waters, and some live in shallow waters. So if we're looking at a sample, we can look at the types of species and their relative abundances to determine what the environmental conditions at that site during this time was. So if we're all clear there, we'll take that and move on to shell middens. For those of you who aren't familiar with shell middens, these are predominantly associated with prehistoric archaeological sites, and you can see two examples here. The one on the left from Maine, from about 4,000 years old, and one on the right, a bit older, from Ireland, 10,000 years old. So these are traditionally interpreted as being refuse pits. So these are from the prehistoric people. They're consuming their shellfish, and they're just discarding the waste. They're discarding all of their shells, and they're accumulating into these thick layers, and you can see across here and all around the woman. So where do Foraminifera fit in? Well, as you can imagine, if we move on to submerged underwater archaeological sites, it may be a bit of an issue to identify what shells were deposited actually during the archaeological period's inhabitation and which shells were just deposited naturally because, well, the site's underwater now. So Foraminifera can help solve this issue. So we have an example, two examples here. The first one from Denmark, we have a site that is dating back to 7,000 years old. And this is the image you see on the left here. Shell middens all on the bottom. And, well, how did they determine if this was an anthropogenic shell midden or a natural deposit? They took a core, such as what Akash had showed in his previous talk, and they actually looked at the Foraminifera within this core. And, lo and behold, they found none within the actual archaeological deposits, as they were interpreted based on the absence of forams, and they only found a single Foraminifera at the surface of the midden. So that would indicate, okay, this is archaeological material. Then if we look at the example down here from Australia, we're seeing, okay, this is on land, but same question. How do we tell natural versus anthropogenic? Well, they took a slightly different approach. And here they quantified Foraminifera from this natural shell deposit on the left and this cultural shell midden on the right, and they found there was a really big difference in the number of Foraminiferas within these two deposits. Whereas the natural shell deposit was really rich, there was really high abundances of Foraminiferas. They found more than 1,000 per 1 gram of sediment. These cultural deposits had significantly less, of less than 50 Foraminiferas per 100 gram sediment. So it's a huge difference, and it was really obvious to differentiate between the natural and anthropogenic. Next, we move on to examples of ceramics. Well, of course, how do we relate Foraminifera to ceramics? Ceramics are so common and so abundant, but people were able to figure out that if we use Foraminifera and identify Foraminifera within these ceramics, well, we can help and deduce what the firing temperatures of these ceramics were. So you see an example in the middle. This is from a site in Iran, and these show several different cooking pots from this archaeological site. So forams are composed of this calcium carbonate, which happens to separate into calcium oxide and carbon dioxide when it's heated to 820 degrees Celsius. So naturally, Foraminifera are not in great condition once heated to this temperature as the calcium carbonate breaks apart. So what this archaeological research did was they looked at Foraminifera within these ceramic pieces and looked at the conditions of the forams. And you can see on the left what the forams look like in the ceramic material. Well, they're a bit broken, a little bit pitted. They don't look so great. But then they compared these to the Foraminifera on the right here. And these are the Foraminifera that are within the natural sediment that they used to make these ceramic materials. Well, they look a lot better. They're very pristine, there's no pitting, and they were able to determine that the ceramic material was actually fired to 750 degrees Celsius through the appearance of these Foraminifera. And lastly, and most common applications of Foraminifera in archaeology are their applications to ancient harbor studies. So ancient harbors can give us a lot of information about both archaeological conditions and paleoenvironmental conditions, and the ability to construct these built man-made harbors provided the option to actually create areas that were protected along otherwise open, exposed areas where ships couldn't really anchor. So we see an example at the top here. This artist reconstruction of Caesarea Maritima in Israel. So this is an example of what was open, exposed coast. Then we see this construction of a suddenly protected, low energy bay. However, this didn't last very long. The harbor ended up destroyed, and we see the return back to the natural, open, exposed conditions. And you can see on the right an aerial image of what this harbor remains look like today. You can see this structure is now submerged. So, how do forams apply? Well, foraminifera assemblages record these different phases in the harbor's construction, its use, and then its destruction, return back to the natural, open, exposed conditions. So, we quickly took this and part of my own research applied this harbor methodology to the investigation of a Hellenistic, which is a 3rd to 1st century BCE harbor in northern Israel at the site of Akko. So as you see here, for those who don't know, Akko on the northern coast, and this harbor was discovered along the southern seawall. And this is the only intact military harbor from this time period, really extensive, but we were focusing specifically for this research on this feature in the middle, this harbor quay. So you can see some examples of the quay here, relatively intact, up to 30 meters in length, and the excavators found that this entire feature and all of the rest of the harbor was underlain by this thick layer of muddy sediment containing abundant archaeological remains. They had broken pottery, they had whole pottery, they had more ornate artifacts. So the question was, well, what was this harbor built on? How was it constructed? Was this built in the marine environment, or was it built on terrestrial land? So we turned to the forams. So as you can see here, a depiction of this harbor quay, with this being the harbor surface that you would stand on, which of course, harbor surface in order to operate has to be 1/2 meter to 1 meter above sea level. Currently, it rests at 1.1 meters below sea level, so significant change. In order to get an idea of the changes in the sediment and the environment during this time and before its construction, we took sediment samples all down the length along the side of the quay and within this muddy sediment layer below. We then compared this to the local sediment. Well, how do the foraminifera abundances change in the local beach sand moving to offshore? And we got that answer on the right. So here we see on the beach, moving to deeper waters, with this axis representing the number of foraminifera. We can see in our shallowest samples, up on the, in the shallow water, we still had abundances of about 2,500 forams, 2,500. Whereas on the beach, we still had about 1,000. So pretty okay. And of course, the forams counts increased as we got deeper offshore. So we compared this to our shallowest samples below the harbor. But these counts were so low, they were only about 700. So this gave us an idea that the harbor was actually probably built on terrestrial rather than in the water itself. And you can see some examples of the foraminifera we identified here. So, in conclusion, this is still a really new application. Foraminifera have been used a couple times in archaeology, but I hope that there's a lot there's more research done in the future, and there's a lot of promising avenues for continued investigations. So, thank you so much.
Okay, are you convinced yet? Okay, the next is a recording. Okay, it's an 8-minute video from Dr. Reinhardt on the work from caves. He's also an Explorer Club fellow. I believe he was even a chapter president in Canada previously.
So we'll be talking about some of our research in the Yucatan. We have worked as well in other parts of the Caribbean, so with colleagues from Canada and the US and Cuba and also in the Bahamas. So all of these caves were also dry, when sea level was low. And because of the porous nature of the limestone, sea level is largely controlling the water level or the aquifer level. So as sea level was rising over the last 12 to 1,000 years, these cave systems went from being dry to being progressively flooded. But this aquifer, certainly for the Yucatan, is the main freshwater source for the population. And the aquifer is very sensitive to climate and sea level change. Freshwater mass gets thicker, and then very close to the coast, because of the flow of that fresh water to the ocean, which then drains out of these coletas or these vents along the coast, we do get some mixing between the fresh and salt water. And so this is something that we're very, very interested in terms of understanding aquifer quality and how it might change with changes in climate. So a video clip that shows both that marine water mass or salt water that's intruding from the coastline, and then the fresh water on top. So the divers are swimming through that fresh water, and as they're swimming through it, they're mixing or causing this mixing between this salt and fresh water, which is giving that kind of oily appearance. But we have very few records of how these two water masses interact. So with that flow to the coast and with increased flow, we can get injection or entrainment of that salt water into the fresh water, and it doesn't take much of that to really affect the quality of the aquifer. So we don't have a lot of instrumental records, certainly in space and sort of time, of this relationship and how it might change with, for instance, increased storm activity for rainfall associated with hurricanes and with eventual sea level change. And this is where our research really kind of fits in, because we're trying to extend that understanding or that record of back in time to of years ago, using sediment cores, and as I'll be talking about, these microfossils, so Foraminifera and testate amoeba. We've been working in this little section kind of close down to the coast, referred to as Yashen. And this is part and parcel of one of the longest underwater cave systems in the world. So this is just not even a current map of the cave passages, and all these cave passages. So currently more than 435 kilometers of map passages have all been done by hand. So this is using compass and bearings and measuring distances. Using divers. So it's a very time-consuming and very laborious process, but we really use and depend upon the cave explorers and the cave divers that are conducting this work, because it does provide us with a really important framework for ultimately interpreting our results. So how are we using Foraminifera and this thing's called testate amoeba? And why are they so useful? So they're basically testate or amoeba-like organisms that produce a shell in a variety of ways, and they're relatively small. So they also produce a mineralized skeleton or shell or test that also then preserves in the sediment record. So it has a fossilization potential. 15, 20 years ago, when we started working in the cave systems in the Yucatan, nobody had really applied them to try and understand cave and kaolin hydrology or change in aquifer conditions. So we're basically taking these tried and true techniques and applications of microfossils and then applying them into the cave environment. So at the heart of what we do, and in terms of being able to go back in time, is we use sediment cores. So here's a couple divers taking some sediment cores in the Yashen cave system, and it's pretty simple. I mean, in terms of technique, we're basically taking a hollow tube. In this case, this is tube that's used for electrical conduit and also central vac systems in houses. And we're basically shoving it into the sediment, and then the sediment is accumulating or getting pushed into the tube. We then remove the tube, and then we have a record of all the layers down below. It certainly goes way beyond any of our instrumental records, and the work that we're doing is very unique in terms of really providing some important records in terms of how that aquifer is actually responding. So in conclusion, I just would like to acknowledge all of our kind of granting support. So we've had support from National Geographic, our Natural Science and Engineering Research Council of Canada funding, so NSERC, as well as Canadian Foundation for Innovation in terms of some of the infrastructure and instrumentation that we use in our labs. Also really have to acknowledge SINDAC, which is a not-for-profit organization in the Yucatan that is promoting exploration and conservation of the Maya aquifer, and they have been an incredible resource in terms of their mapping and their exploration efforts and gaining information in terms of where we can collect samples and how we can collect samples, as well as also providing support in terms of the actual field work. So helping out with actually collecting samples, getting access to certain caves, and being able to also conduct the work. So without SINDAC, most of this work would not be possible. Well, thank you very much. I appreciate and being involved in this, and I hope that this was interesting and gave a slightly different perspective on the use of foraminifera and microfossils, unique application into caves and cave sediments. Thank you very much.
Okay. So, I know you've been waiting your whole lives to ask questions about foraminifera. This is your chance.
I really do not still. I still don't really understand. Could you put it in really simplistic terms? How you can predict which corals are healthy through foraminifera?
Sure. I mean, this is really for Akos.
Yeah. So just please, dumb it down.
The two sentence. Well, we had a long discussion about this. So do you want me to give my translation of it, or yeah?
Then I can take it over.
So we had this discussion, which I definitely agree. Akos didn't quite, I think.
I get a lot of stuff.
Yeah. So I'll summarize it in my simplified version. So basically, the idea is that it's possible, because you can do you can get information about what the background environments are for these places. So the idea is that in combination with coral monitoring, okay, it's not a standalone, but what the forams, by looking at what that study figured out, is that you don't have to look at all 41 genre. You can tell, just looking at the proportions of 3 of them, whether or not it's a place that naturally has a lot of transition, or if it's somewhere that's been altered, and so therefore you could stop doing something, or you could fix something to make a change. The change would be more dramatic, like the delta, the potential of investment in a place where it's responding to artificial changes in the environment versus a place that is going to have a fluctuation that even if you invest a lot of time in, let's say, in the mechanical sense of like cleaning out the macroalgae, if that's the natural environment, it's going, it's a little bit, it's more Sisyphean than if you do it in a place where the changes are because it caused by a change to the environment rather than a changing environment that is naturally changing. Did I make it more confusing?
I like it.
Essentially, yeah. What is the background like norm for the area? If it is it's change, if it has a lot of fluctuation over time, and you can see that in the mix of the foraminifera that are there, then it's going to change. You're kind of fighting against what is naturally happening. But if the change is because of an acute anthropogenic influence on it, then there's humans would have more control over whether or not that intervention would be helpful. This is really a decision-making thing, like where to invest more.
Yes. The forams, they don't know why they are suffering. They just don't feel good themselves. And one of the most advantageous part of using the forams is that they respond in much shorter time than the corals respond to any kind of anthropogenic influences. So the lifespan of the forams, the two ones that were pulling to the coral direction, they can live up to 2 years, and the other one can live up to like only a few months. So if we see changes already, we can recognize and capture it in the moment of when the transition begins. Those are the best candidates for restoration efforts. On the other hand, when we can see only in these 3 genera that it's pulled more like with a quinquiloculina direction, then we know that they are suffering for a little bit longer time than just recently shifting coral reefs. So they have better chances, higher probabilities to survive and then come back from this macroalgal phase shift.
You kind of hit a little bit on one of my questions as a follow-up to that. I realize this is a new tool, burgeoning science, to use for the change in the climate. What span are we talking about? As humans, we fail to plan long-term. Would this be a tool that you could use in like a 5-year increment? You kind of answered with the 2-year life cycle, but what is the practical segment of time that becomes useful to start recording the change so we can apply it?
So we start with taking a surface sediment sample, and these coral reef ecosystems, they are composed of many parts, and usually in these regions bioturbation also plays a role. So when we take a scoop of the surface of the sediment, then we usually take a time averaged, we call a time averaged foraminifera abundances, which can go back to, let's say, 100 years. So this is a big time average, and therefore this foraminifera approach, rather than just replacing all the tools that we have for coral reef monitoring, it's more like an addition to that.
Although actually, there's a different study. The sections that you see can be produced in a 24-hour period. They're very rapid. So we have done studies where we're able to measure sort of chamber by chamber. And for example, we were able to recognize a flood that brought in more heavy metals. So in a single, by analyzing, if you're interested, I can later, refer you to the academic paper on it, but basically by measuring the chambers, we were really surprised that we actually saw the peak from a flood that brought in certain pollutants. So it is something that you can use even on a monthly, use for monitoring on very short scales and recent. And you notice that some of them were pink. You saw the pink coloring? That's an artificial stain that is used to figure out which ones are living and which ones are dead when you collect them. So let's say an event happens a month ago and we go and collect, we can stain it. And then if we want to study ones that lived through or, that are alive at the time of collection, we'll select those so that we can, like tree rings, basically, like go back from the most recently produced chamber, and then that way we can count back through the chambers and get some sense of time. But those are on very short time scales. Oh yeah, it can be done on days. I mean, literally, and some of them with, this is still being developed, but we're definitely using it on. We can see a day, the days of the flood. Personally.
How sensitive are they to acidity? Can they be used
They're
in-
just like coral. Oh, sorry.
Right.
Many of them are very much like coral. I mean, there are different types. So the ones that are calcium carbonate, and then there's also agglutinated type. So we didn't go into all the different and organic walled, but the majority of them are calcium carbonate. And so just like coral, they can bleach if they have symbionts. So research in the laboratories, I don't actually know of any natural bleaching experiments with foraminifera, but they might exist. But in the lab, bleaching, they do respond to acidity because they can't produce, and in the laboratories, way before a lot of the research on acidification in corals, micropaleontologists were, I say, torturing forams regularly by dissolving out the shell and just working with the cell. And the cell's living and reproducing and doing all the things it does, but not producing a skeleton. And this is a whole nother session, but some coral can do that as well. They can live through the loss of their skeleton. They can reproduce as non-skeletal coral, and they actually lose their colony, like the polyps will become individual polyps, look like an anemone, basically. And then when the conditions go back to non-dissolving, basically, they will rejoin their colonies and regrow.
In Vieques, I'd just like to add one sentence to that. In Vieques, at the bombing site, we can see especially like the tests with the being suffered from higher acidity. And those tests look a little bit like holes, like a moon, surface of the moon. And again, we observe it and we make a count how many of them look a little bit suffered from acidity and how many are pristine. And we have high numbers of, let's say, abraded foraminifera.
All right, thank you very much. We're going to have to wrap it up there. If the three of you will stand just in, I'm sorry, did you have something else you wanted to add?
No,
Okay.
You'll have to ask a list of questions later about the archaeological applications. We do have one more video, though. Should I come up to the podium? Sure. Okay, all right. I don't need this. 1 minute, 1.5 minutes. Okay, just a couple of comments before we break. So downstairs, there in the Apollo room, if you want to go and see for yourself, we have, thank you to Martha Shaw, who really helped organize, and the rest of the committee for the Oceans Week. We have microscopes, and I've forgotten who generously brought them. I forgot the name of who. Trinity School generously brought these microscopes. So please go in there. There's a whole bunch of samples, and you can have a look yourself at what they look like. And you'll see a video, which is going to go through, some of them are more artistic, but you'll see on some of the screens for the next hour. I do want to end, though, I'm going to shift a little bit because I am part of the Middle East chapter for the Explorers Club, and we're about to we established the chapter only 2 years ago with Hamish Harding. And so the members of the chapter, who I'm the only one here representing this time, each wanted to put together a very short, just 90-second video, in his honor. So I'd like to play that.
One last thing, if I could get the team to stand back up here and face the audience. We've got one little